Yeah, so well, I really don't have to give any talk because Libik has said it all. There has been an incredible amount of data coming out in just the last year, that both from the clinical side but also from the more basic translational side that I am to highlight. That is really hard to actually capture everybody's work. But now we went black instead. So when I started in the lab at NIH doing my training, I felt a little like Sisyphus, just like you probably feel more like when you get the diagnosis. There was nothing in 2006-07 when also the Cholangiocarcinoma Foundation started, as Stacy said, not even a standard of care. We had no cell lines really. We had no primary cell lines, no organoids didn't even exist. And getting tissues to do any omics, that's what I represent in science, a lot of omics work, but really, really difficult to get your hands on and took a long time. So I took this very boring but quite important statistics from PubMed. You can all find it, it's very easy. Put cholangiocarcinoma in as a search term. Look at the year that we're supposed to update, 2021. And since 1949 until 2022, 73 years, there's been published around 18,000 papers. And last year alone, nearly 2,000 papers were published, that's 11%. So when we have a 4-time increase or 3.5-time increase in clinical trials, we have an 800% increase in publications. Now, you can always publish. So it matters if the publications are any good. And I hope I will convince you that in cholangiocarcinoma, maybe not all 2,000 papers, but a lot of them really will make a difference, and also will make a difference to what Libika and all her colleagues in the clinics, and what you will see in the future in the next 5 to 10 years actually has come out already in 2021. Besides that, it is a great year to stand here and present updates to cholangiocarcinoma, having not even 2, but actually 3. I'm sorry that I forgot QEDs and insights. Helsing's ifigratinib also was approved actually, but we in 2020 had FGFR2 fusion, penigatinib from Insight. Last year, Tipsovo against IDH1 mutant from CVA, and then also ifigratinib, that should have been on the slide of course, against the FGFR1 to 3 alterations. Besides that, is an even more great opportunity for me to stand here and be invited to co-host the meeting in 2022. It's 10 years after the first genomic classification of cholangiocarcinoma patients came out on my own hands from NIH, building a genomic classification, prognostic classifier, which actually still stands today, indicating that the poor prognosis group had inflammation and proliferative high capacity, high degree of EDFR, HER2 unmet overexpression, and the KRAS mutants. And in the better prognosis, patients surviving longer than 50, more than 50% patients surviving longer than 5 years, having changes in genes involved in immune response. Now, very shortly after we started getting in from the dark ages into where everybody else in oncology was already getting into next generation sequencing approaches, and I just highlighted a few of them. There are many, many more that have come and are still coming out. We try to supervise classes of patients, and I'm sorry to talk about living beings as classes and as objects, but that is how we need to see it in grouping things and not as the individual. So we identified IDH, KRAS, TP53, and at the time patients we called undetermined because there was not really any mutations, or it was very heterogeneous in that population, and they did not have IDH, KRAS, and TP53 as drivers. During that time, FGFR2 fusions became highlighted, and the therapy is now approved, as we just went through. So FGFR2 fusions actually turned out to be in the undetermined group. But that's a different kind of alteration than mutations. you see that when you look at the RNA, so it's a fusion between two proteins. There are also, which I hope will come in the next few years, an important amplification in a gene called metal-13, and more to come on that. Now the cohorts went up to nearly 500, and only intrahepatic cholangiocarcinoma, so not just all comers, extrahepatic gallbladder and so forth. In this study, we also started looking at drug responses to find something off the shelf that could be used potentially in a clinical trial setting, or one could try to give in these specific patient groups. There was also clinical importance to these groups, of course, and some of this work still needs to be validated. Now, we are in 2022, just a few years later, and we are a triple number of patients, nearly 1,500 patients going in a rare cancer. That's really amazing. The class still stands. A cluster 1 with KRAS, TP53, cluster 2 with IDS and FGFR2 fusions, and a cluster 3 that still has nearly 50% of all the cases that are somewhat undetermined what really happens in their genomes of these tumors. But there are a lot of chromatin remodeling factors, and I think that could be interesting for the future as well. Now, work has also moved on from just integrating different types of genomic data set, but also integrating the pathology. So here they integrated if the patient's tumors were from the large bile duct area or from small bile ducts, and that matters to what mutations you also find in there find in the DNA, and therefore also what subgroup they should belong to. Then last year, or maybe a little before last year, but anyway, cholangiocarcinoma also moved into the area of single-cell genomics, and now becomes a little long-haired maybe for a lot of you, but it actually leads to some of the important stuff that Vibeke pointed out. For example, with the immunotherapy, we can start to understand why some will respond and some will not respond, because it gives an intensified view of the stromal and immune cell niche, the desmoplastic tissue type that you might have heard some of the clinicians talk about when you are diagnosed, how much stromal tissue is there around and integrating into the epithelial part of the tumor. When we talk about the mutation space, it's always in the epithelial part of the tumor and not so much in the stromal and immune space. But that interaction between the stromal and immune niche with the epithelial cells are very important for the therapy and the way that you will be treated in the future. So these studies started to highlight things we were interested in already before single-cell genomics, but we were not able to really study in details before now. Highlighting the microenvironment, particularly the cancer-associated fibroblasts, the TAMs, the tumor-associated macrophages, and the T cells in which immuno-oncology currently is most focused on, the CD8 cells, and another cell type or T cell type called mite cells that you will hear about later in the conference as well. These are again just a few of the graphical abstracts from these papers. There are many, many more. We also got into proteogenomics. So what does that mean? That means that we are looking at the proteins, the workhorse of what goes on in your cell and how the cell will behave. That comes after the genes and comes after the DNA. So what is actually being made and what is making the difference phenotypically of these cells, as well as the metabolome also will be part of this. And in this study, they highlighted the different groups that we have already discussed, and things are changed in the proteogenomic landscape of a KRAS patient, more the EMC, so the stromal area and local adhesion, how cells are interacting with each other, as well as in the IDS and BAP1 mutant cases where a lot of bile acid changes are occurring. Oops, can I go one back? So they made a classification based on the proteome for drug response in the landscape of these patients, calling them S1 to S4. S1 is the inflammatory, KRAS mutant. We have known that back from 10 years back when we just looked at the genes. S2 is mesenchymal, so that's the CAFs, the cancer-associated fibroblasts, the stromal tissue, and lymph node metastasis that they highlighted with. S3 is the metabolic, the P53 mutant cases, metabolic changes, and very highlighted for HPV positive cases, so hepatitis B virus infection. and S4 is differentiated, so the cells are differentiating into a different phenotype, IDS mutant, BAP mutant, FGFR2 fusions, and adhesion between the cell types again, and bile duct specific. Here, the biggest change from our previous classification are probably that the p53 mutations are correlated with a metabolic phenotype, which was, to me at least, more alike to the IDH mutant and BAP1 mutant cases in the past classifications. But we will see if this stands. This paper, which is an amazing paper and a really long weekend job to read if you go into it to try to understand. There's so much data. Also went into metagenomics, so looking at the different bacteria in your gut and how that communicate with the liver, so the liver-gut axis in line of what type of mutations there are in these tissues. And again, they can classify the patients into 4 different groups, the same groups as before, but having hugely different bacterial component, different microbiota compared to the other groups. So the proteogenomic subgroups have distinct tumor microbiota diversity. Bacteria composition and function actually changes, re-educates the immune and metabolic rewiring of these tumors. This is something that are still in the really early stages, and we will hear more about this as well from Tim Gretton later in the conference. Very shortly on the stromal list, this is, I think, quite important, but I'm not going to go deep into it. We're going to hear more about this as well at the conference. So cancer-associated fibroblast or the stromal list is just not one community. It's not just one area. It's just like the epithelial cells, very different subpopulations. And in this study of some of the different studies that have come out, you have 4 different subclasses of calves. Some that are more inflammatory, some that are more mesenchymal and so forth. And some of them are seen, in this case, the mice calves and the inflammatory calves, to actually both in mouse models as well as in human intrahepatic cholangiocarcinoma to promote the disease. This is quite important in line of something that the community has wanted to do, and I at least was quite interested in doing for a long time, was to deplete the stromal tissue so you would get more access to the epithelial tumor cell for the therapy to easier target and kill the cancer cells. This was done when thinking about doing this in cholangiocarcinoma, in pancreatic cancer, a tumor type that is as bad as cholangiocarcinoma and is very alike to cholangiocarcinoma in many ways, both clinically and pathologically, and also in many ways on genomics. They depleted by targeting the hedgehog pathway, and when they went to clinical trial, it was detrimental. So it was all killed as an idea. Maybe because one has to target specific calves. I'll jump over this one. This is a paper that I have not even yet been able to fathom myself. It's a humongous paper that just came out, and I needed to highlight it for one reason and one reason only. Cholangiocarcinoma, gallbladder, is included in this study. It would not have been 5 years ago. They didn't see anything specifically for cholangiocarcinoma or gallbladder, unfortunately, but it's a 25,000 people study with nearly 100,000 metastases included in the study, so it's gigantic. And I think when we start doing post-hoc analysis, so pulling out specific data we might be interested in, and in my lab it would be, of course, the cholangio patients and the gallbladder patients. When we start pulling those out from this very big study, we might actually see something useful for your disease as well. Now, talking about all of the omics and the importance of mutations matters, now updated to biomarker matters. I agree biomarker matters, because if we really are to make a difference in cholangiocarcinoma, we need to diagnose the disease much earlier than we do today. And for that, biomarkers matters, diagnostic biomarkers, but there are many different biomarkers out there. There are also prognostic biomarkers and biomarkers to monitor both the development of the disease, but also monitor the response to therapy. And nonetheless, is it actually a good idea to continue the treatment, or should one go over to another treatment or stop the treatment? A few years ago, this study came out in Nature. I think it might have flown over most of the cholangio community, but actually bile duct was included. This is the DELFI study from Victor Velasco, John Hopkins, where they sequenced a lot of patients, their cell-free or tumor-specific DNA, to see if they could diagnose, define the different disease types. And I highlighted in red the bile duct tumors, and I think it is with a specificity of 88%. So it actually shows the potential to screen populations or monitor populations for an increase in certain amount of cell-free tumor DNA in the serum, not doing any tissue biopsies or anything like that, simply just a blood sample, and you can test if there is a risk of having cholangiocarcinoma. Now, you could also use that as highlighted by Lippica, being done by mutation and others for looking at specific mutations in the serum to see what type of cholangiocarcinoma you have and if you're responding to the therapy. We published a few years ago a study on 450 patients, advanced patients, looking at the serum and looking specifically at IL-6. I don't want to go through this work diagram. That's not really important. The importance were that we compared this to CA19-9, which we believe is not really the best diagnostic marker in the world. IL-6 actually was significantly better, both seen in the validation cohort for predicting death over 1 year, as well as our ability to establish or estimating a biomarker threshold level of IL-6. If you look at the right-hand side, on the continuous AUC, so the area under the curve of predicting survival, you can see that the red and the blue curves for IL-6 and another marker in the IL-6 pathway called YKL40 is significantly better at predicting the progression of the disease. We had longitudinal samples, I should say, compared to CA19-9. This is not important, but this leads to basically concluding the fact that besides the success of durvalumab targeting as an immuno-oncology drug targeting the interaction between the T cells and the epithelial cells, targeting specifically as a targeted therapy FGFR2 fusions or IDH mutant cases, or many of the other things that Lippica also highlighted, single cell genomics might be highlighting for the near future also the potential of other types of immuno-oncology that are not necessarily targeting the interaction between T cells and the epithelial cells, re-educating the T cells to kill the epithelial tumor cells, but actually in myeloid lineage such as the calves or the fibroblasts or the TAMs, and targeting interaction between these cells. So another type or subversion of immuno-oncology, if you will. That could be targeting the IL-6. IL-6 is produced typically by the macrophages, the tumor-associated macrophages, and the receptor is on the epithelial cells. So there's a potential for a drug called tocilizumab or others in that line. And more targeted specifically, there would be a future for targeting areas of the SWIFT-NU complex. BAP1 mutations are now back again as nearly a subgroup by itself, and could be an interesting area to investigate for new therapies. And then the revisiting of receptor tyrosine kinases is also highlighted. It kind of died 10 years ago with a lot of failure in clinical trials, but if we now find the right patients, maybe we can also reuse some of those drugs that are already out there. Thank you very much.